friends, how are you? Hope all are fine. Welcome to my channel, Sam Biology Class. Today we will discuss about sandwich Elisa. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. Sandwich ELISA is a type of enzyme-linked immunosorbent assay that is used to detect as well as quantify antigen in a sample. In this assay, the antigen is immobilized as well as detected indirectly. Compared to direct and indirect ELISA, this technique is more sensitive because the antigen is detected by two different antibodies. In direct and indirect ELISA, the antigen is immobilized by direct adsorption at the bottom of the well of the microtiter plate. By contrast, in sandwich ELISA, the antigen is immobilized indirectly by using a capture antibody. These are the different steps of sandwich ELISA. First step is plate coating. In this step, the well of the ELISA plate is coated with a capture antibody. After coating the well with antibody, the plate is incubated in order to allow the capture antibody to bind to the bottom of the well. In our example, there are two capture antibody that binds to the bottom of the well of the microtiter plate. These are the two capture antibody. After incubation, unbound antibody are removed from the well by washing the plate. After all unbound capture antibody are removed from the well, the plate is blocked by adding a blocking buffer that contains a irrelevant protein such as bovine serum albumin. Blocking is necessary to prevent the non-specific binding of either antigen or antibody to the surface of the well. If the plate is not blocked, then either antigen or antibody will bind non-specifically to the surface of the well, and this may give rise to false positive. Blocking is followed by antigen addition and incubation. In this step, a sample suspected to have the target antigen is added to the well and incubated. If the sample contains the target antigen, then during incubation, there will be binding between the target antigen and the capture antibody. As a result, antigen-antibody complex will be formed. In this case, the result will be positive. These are the two target antigens that are attached to the capture antibody. If the sample do not contain the antigen, then there will be no binding and the result will be negative. After incubation, unbound antigens are removed from the well by washing the plate with phosphate buffered saline. Next step is detection antibody addition and incubation. In this step, a solution containing detection antibody is added to the well and incubated. During incubation, the detection antibody will bind to a distinct epitope of the same antigen. In our example, these are the two detection antibody that binds to a distinct epitope of the same antigen. So the antigen used in sandwich ELISA must have at least two distinct epitopes for binding with two different antibodies. Because in this assay, the antigen is sandwiched between two different antibodies, the name is sandwich ELISA. After incubation with detection antibody, all unbound antibody are removed from the well by washing the plate. In one type of sandwich ELISA, the detection antibody is directly linked with an enzyme. If this is the case, then there is no need of adding secondary antibody. And in this case, after all unbound detection antibody are removed from the well, when a chromogenic substrate is added, it is converted into a color product. The amount of product formed is proportional to the concentration of antigen in the sample. Such type of sandwich ELISA in which the detection antibody is directly linked with the enzyme is called direct sandwich ELISA. Direct sandwich ELISA. In another type of sandwich ELISA, the detection antibody is not linked with the enzyme. So here for detection, an enzyme linked secondary antibody is added to the well and incubated. The enzyme linked secondary antibody will bind to the detection antibody during incubation. So these are the enzyme linked secondary antibody that binds with the detection antibody. After all unbound antibody are washed out from the well, a chromogenic substrate is added which is converted into a colored product. And the amount of product formed is proportional to the concentration of antigen in the sample. Such type of sandwich ELISA in which additional enzyme-linked secondary antibody is added for detection is called indirect sandwich ELISA. In order to determine the concentration of antigen in the sample, a standard calibration curve is prepared. For this, a series of standard solutions containing increasing known concentration of antigen are taken in different test tubes. In one microtiter plate, about 10 wells are marked as standards, one as blank, and another well is marked as samples. All these wells were first coated with the capture antibody. After coating the wells with capture antibody, the plate is incubated, after which unbound capture antibody are removed from all the wells by washing the plate. In next step, the wells are blocked by adding a buffer containing bovine serum albumin. After blocking, the standard solutions containing different known concentration of antigen are added to the wells that are marked as standards. In this well, no antigen is taken. And in this well, the sample suspected to have the antigen of interest is added and incubated. During incubation, the antigens will bind to the capture antibody that are immobilized at the bottom of the well. After incubation, unbound antigens are removed from the well by washing the plate. This is followed by detection antibody addition and incubation. In sandwich ELISA, the detection antibody is directly linked with an enzyme. So in this assay, the solution containing the enzyme-linked detection antibody is added to each well and then incubated. During incubation, the detection antibody will bind to the target antigen at a distinct epitope. After binding between the enzyme-linked detection antibody and the antigen, unbound antibody are removed from the well by washing the plate, after which a chromogenic substrate is added. This substrate is converted into a color product. By contrast, in indirect sandwich ELISA, the detection antibody is not linked with the enzyme, and hence, after binding between detection antibody and the target antigen, another enzyme-linked secondary antibody is added to all the wells. The secondary antibody will bind to the detection antibody. After incubation, unbound enzyme-linked secondary antibody are removed from the well by washing the plate with phosphate buffer saline, following which the substrate is added. The substrate is converted into a detectable product, which is then measured by a plate reader. After development of colors, the optical density of all these wells is then measured by a plate reader. After measurement of optical density or absorbance, a standard curve is prepared by taking concentration of different standards on x-axis and their optical density or absorbance on y-axis. After preparation of the standard curve, the concentration of antigen in the sample is determined by interpolation. Friends, this is all about sandwich ELISA. For more updates, subscribe my YouTube channel, Sam Biology Class.